All right. So now, what we are saying here is that we have these phases. Then comes the uterine phase. So these things are connected. When you have the follicular phase, on the uterus you are having the proliferative phase. So they are happening exactly at the same time. When you are having the luteal phase on the ovary, you are having a secretory phase in the uterus. So they are connected, as we shall see. And then we have menstruation, which is from day 8 to 0th day, roughly. Now, let me emphasize on one thing, that between the ovarian cycle and the uterine cycle, the most important cycle for you, clinicians, is the ovarian cycle. That's where reproductive physiology is. The uterine cycle is useful for histology purposes. Okay? But it's a small component, very small component, as we shall see. But the ovarian cycle is what you need to understand. This one. Okay, so now let's start the story. Remember, we said that when the primary oocytes are produced during that period of embryology, when oogonia switches to go into meiosis, right? We said the name changes from oogonia to primary oocytes. And you said those primary oocytes are never exposed. They will soon be covered with the flattened cells known as the theca cells. And the combination of the flattened cells and the primary oocyte, that 1 to the power 0 is primary oocyte, is called the primordial follicle. So the primordial follicle is a combination of the primary oocytes and the theca cells. If this primary oocyte is not completely covered by the theca cells, it's dangerous. That cell will die. It becomes aquatic. That explains why, from that huge number, you end up with a drop of 1 to 2 million at birth. Because some of them are not thecally covered. Now, look at this. From about 85 days, for 85 days, there's some process which happens in the ovarian cortex, which takes about 0 to 85 days to move the primordial follicle from here to the next level, known as the preantral follicle. So this change from here to there is just a change in size. But there is no fluid anywhere. No wonder it's called preantral, before fluid. Antral is fluid. So this process takes about almost 3 months. However, what is funny is that once a girl reaches puberty, about 300 of these primordial follicles will be recruited per month to undergo further development. As if we have picked you, Dr. Simpugetsi, you are going for further training to do PhD. Okay? So these will be recruited. Now, what will be happening is that from primordial to preantral stage, and take note, there's no change in the primary oocyte. It remains primary, it does not change. What is changing is the outer layer. You get the point? The outer layer is what is changing. No wonder the names are changing as follicles rather than the oocyte. So don't confuse the two. Now, the growth from preantral to antral, this one going this way, is hormone dependent. From primordial to preantral is non-hormone dependent. And that's why it takes about 85 days. But immediately you reach preantral, going this way, then hormones are involved. And those hormones are expounded by what is known as the 2-cell 2-gonadotropin hypothesis. 2-cell 2-gonadotropin hypothesis. We sometimes call it the central dogma of reproductive physiology. She's so much a dogma. So now let's see the story. Here we have theca cells, here we've got theca cells. Then all of a sudden we have got fluid. And then the primary oocyte remains in the center. You see that? Very soon there is a layer of glycoprotein which is formed around the primary oocyte, and that layer is known as the zona pellucida. zona pellucida. Between the cell membrane of the primary oocyte and the zona pellucida is a space known as the perivitelline space. This space is important because that's where you have storage of the polar bodies. So polar bodies are extruded into the perivitelline space. And the cell membrane of the primary oocyte is sometimes called the ulema. Remember, sarcolemma, ulema, and so on. Then comes the fluid. The fluid is known as the antral fluid. Now, imagine these theca cells on the outside will divide into two layers, two distinct layers. There will be what are called the theca externa and the theca interna. And then comes another layer which wasn't there of simple cuboidal cells known as the granulosa cells. So the granulosa cells are inside and the theca cells are outside. For you to understand how this arrangement comes about, you need to go back into embryology of the ovaries. But we don't have energy for that. Now, look at this. Now, what will happen next is that since now we are saying this is hormone dependent, the brain has got hypothalamus. And the hypothalamus will release a hormone known as gonadotrophin releasing hormone. So GnRH, which is a very special hormone because it is released in pulses. So it has got an amplitude and a frequency. And once you lose the amplitude and frequency, it explains partly what happens in Stein-Leventhal syndrome, also known as polycystic ovarian syndrome. It can be very nasty, loss of pulsatility. No wonder, again, you can use GnRH analogs to shrink a fibroid, just like you can use the GnRH antagonists to shrink a fibroid. So it's very rare that an agonist and antagonist will lead to the same. But that will be a discussion for another day. So GnRH will then act on the anterior pituitary gland to release two hormones known as luteinizing hormone and follicle-stimulating hormone. Now, here, follow the story very closely. What luteinizing hormone will do first is that it will convert cholesterol, which is circulating in blood, it will convert it into androgens. Cholesterol will be converted into androgens. These androgens are bad chemicals. A girl should not have androgens, otherwise they'll start looking like me. Right? So the androgens like DHEA, known as dihydroepiandrosterone, we also have androstenedione and testosterone. So a woman does not want to have this. So these androgens will be quickly converted. Upon realizing that I've got bad chemicals, they will be converted into estrogens. And again, estrogens have got three types. There's estron, there's estradiol, there's also estriol. So there's E1, E2, E3, but the most potent is E2, estradiol. There's even E4, estretol. You can go and read on it and what it does. Now, this conversion of cholesterol to androgens is by the luteinizing hormone, right? And it happens in theca interna cells. Whereas the conversion of androgens to estrogen is not by luteinizing hormone, but is by follicle-stimulating hormone. And it's not in theca cells, but it's in granulosa cells. So that's why we are saying two-cell, two-gonadotropin hypothesis. Two different cells and two different hormones. And the last stage of converting androgens into estrogens is known as aromatization. So this is how drugs like letrozole work. Drugs like letrozole, which is used for ovulation induction in somebody who has got subfertility, for instance, instead of using clomiphene, you can use letrozole, for instance, is that you are going to block aromatization so that you drop the estrogen levels deliberately. And when you drop the estrogen levels, they will make a negative feedback, not on LH, but on FSH. Because what you need for somebody to eventually have a child is more of FSH than LH. So that is, again, a discussion for another day. I just wanted to say that the menstrual cycle is an application of so many principles in gynecology. Now, these are produced. Now let's look at what does the estrogen do. This is very important. What do this estrogen do in the body of a girl? The first thing that they will do is that they will induce progesterone receptors in endometrium. They will induce progesterone receptors in endometrium. This is called priming. It's called priming. So what it means is that the estrogen produced by these follicles is going to visit the endometrium. And in the cells of the columnar epithelium of the endometrium, it will undergo processes like transcription, translation, and one of the proteins that will be produced is the progesterone receptor. So it is estrogen which produces progesterone. You get the point? So if this endometrium was never visited by estrogen, then it will never have progesterone receptors. Such that in future, even if we brought progesterone on this endometrium, it will not work. Because the receptors or the magnets for progesterone are produced by estrogen. And that is the principle behind what is known as the progestogen withdrawal tests. Somebody has stopped having menses, wants to see what is wrong with this endometrium. Why is it not shedding off to give us a mens? Let us give progesterone for 10 to 14 days and stop. And see whether it will respond as it normally responds. You get the point? So that's the principle behind the progestogen withdrawal test. Again, another principle which I'll go in detail the time we discuss amenorrhea. So what I was trying to say is that estrogen goes and acts on a receptor within the columnar epithelium. So it's as if the columnar epithelium already has a receptor for estrogen. It's on the nucleus. Then that nucleus induces a process to produce a protein, which is the progesterone receptor, which will be on standby awaiting progesterone later on in the process. Okay, that is just one. The second one is now, before I even go to the second effect, the problem of estrogen working here and never to be visited by progesterone is that it risks this